Okay. Well, thank you for coming, and thanks people on Zoom. Our speaker today is Jim Strother, who is assistant professor of biology, and he is working at the Whitney Marine Lab, but he's in our department, in the biology department. Jim received his bachelor's degree from UC Berkeley in physics, and then went to UC Irvine for a PhD in biology, studying fish respiration. And from there, he did postdoc work at Howard Hughes Medical Institute, where he worked on another model organism, namely Drosophila. And then he was hired as an assistant professor at Oregon State, and he spent several years there before coming to UF in 2019. So his current research focuses on questions at the interface between neurobiology and physiology, using fishes as model organisms. And he's especially interested in understanding processes at multiple levels, like from cells all the way up to individuals. And his current projects include studies on the nervous system, cardiovascular system, and the effects of stress on behavior in this model organism. And so today he will be telling us about zebrafish.
All right. Yeah, thanks for joining me today. I'm happy to have the chance to talk to you a little bit about zebrafish as a model of system. I had a lot of fun actually putting the slides together. It was an opportunity for me to go back through a little bit of the history of the system and kind of read some of the original papers. So I hope that you enjoy it as much as I enjoyed putting it together. So the history of zebrafish really starts in the 1930s with a paper by Charles Kreisser in 1934. Kreisser proposed that zebrafish would make a great model system for trying to understand the development of animals. And what's remarkable about this paper is how similar his conclusions are to the conclusions and justifications that we would use today. He puts together a sort of list of these are the advantages of the system. And it's exactly the same list of advantages that I would put together. He points out that there are small tropical freshwater fish that's native to India. It was imported to Europe and then imported from Europe to the United States in I think it was 1905 or thereabouts. And so by the time that he wrote this paper in the 1934, they were sort of common throughout the aquarium industry. They'll breed very readily under laboratory, under sort of hobbyist aquarium conditions. That's very different than most animals. If anyone has experience trying to rear animals, most animals will only spawn when the conditions are perfect, when the offspring have the greatest chance of survival. There's certain cues that the animals might look to in terms of day length, about which would reflect seasonality. Lots of different factors would go into inducing spawning. Whereas with the zebrafish, there are factors that are important for spawning, but they're fairly loose. Even in a plastic tank with a little bit of vegetation, the animals will readily spawn all year round. When they do spawn, they produce hundreds of eggs in a given pairing. And that's hundreds of different embryos that you can then look at. So lots of different opportunities to look at the effects of different mutations or genetic manipulations. The eggs are externally fertilized. So if you were to look at an organism that would be internally fertilized, such as a human, a lot of the interesting development happens when the developing embryo is completely inaccessible. Or if it's inside of an egg, like a reptilian system, you might have to open up the egg in order to see some of the early developmental stages. Whereas with these animals, the eggs are externally fertilized, and so that's you can watch development from the very first cell division onwards. And you can also manipulate that system, so you can get unfertilized eggs and perform different manipulations that way as well. They're also relatively small and transparent, so you can see some of the early development as it occurs. So I put together, or I found a video that shows you what that looks like. So this is this is a just fertilized zebrafish embryo. So it's surrounded by the chorion. The actual embryo is here. At this stage, it's a single cell, which is up at the top. Zebrafish and other fishes have it's called teleolesethal development. So unlike in something like a frog, where the entire embryo is going to undergo cleavage, and so you might have one big yolky cell that's then going to divide. In our case, the cell is up at the top. The rest of it is a yolk, and it's this cell which is going to undergo cleavage and eventually turn into the animal. This yolk sac is going to sort of remain as a separate part of the animal. So I'll let you watch that happen, or let that unfold. So that's single cell. And then you'll start to see the little furrow there. Division, cleavage, another cleavage. Each one of those cleavage events takes about 15 minutes. So it's sped up quite a bit. And the entire development that we'll see takes around 3 days. See the massive embryos, and then all of a sudden, out of nowhere, is what looks like a fish. There's its eye. The tail. Just to wiggle around a little bit. And then at 3 days, it'll hatch out of the chorion. And around 4 days, it'll inflate the swim bladder and start swimming around. So at around 5 days, they're more or less fully self-sufficient. Well, they're self-sufficient from when they're fertilized, but they're feeding and moving about and sort of taking care of themselves after around 5 days. Navigating through complex environments, avoiding predators, finding food items, capturing the food items, ingesting them. They're fully functional little organisms after 5 days. So you'd think like this is the point where zebrafish sort of explode as a model system. But in reality, what happened is this sort of paper came out. There were a lot of advantages to the system. But there are similar arguments could be made for a lot of other systems as well. And so what happened is this sort of nice paper was published in 1934, and different people tried zebrafish here and there for a little while, but it didn't really take off. There were other model systems with similar sorts of advantages, like Xenopus. And so it sort of like kind of simmered in the background for a little while. Until it was picked up again by George Streisinger, who really took the system to the next level. And he has a really sort of interesting history, which kind of plays into the development of the model. So he was born in Hungary in 1927. His family fled from Nazi persecution, and they arrived in New York. What's really phenomenal, and I only learned this in the course of putting this together, this talk, was that he, in high school, worked with Theodosius Dobzhansky on fruit flies. I misspelled his name, but Dobzhansky, if you went to the fruit fly model systems talk, was really seminal in establishing fruit flies as this sort of incredible system for doing genetics work. And so in high school, he was working with Dobzhansky. which is pretty phenomenal. And was actually publishing papers from that period. He went on to get his bachelor's from Cornell in 1950, another typo, sorry, before going on to do work in the Luria lab. This was really the period where DNA had been identified. This was just after DNA had been identified as genetic material, just before the sort of structure was being determined by Watson, Crick, and Franklin. And there was a really sort of active race to understand how DNA as a heritable material was operating. What was the relationship between DNA and RNA and protein? And so the approach that he was taking in the Luria lab was work on phages, viruses that infect bacteria. And they were using these phages to understand, again, this relationship between DNA and RNA and protein. Manipulations like removing different types of material from the phages and looking at how that affected the bacteria, trying to make mutations within the phages. Yeah. So, sorry. Yeah. So this is 50. So there's two typos here. I think they're the only two typos in the talk. One is that's not how you spell Dobzhansky, and that's 1950. So then he continued to work on phages for his PhD. He started his postdoc, and with his postdoc, he was trying to take some of these insights and apply them to understanding how genetics worked in other organisms. And he focused on plants, and so he spent a lot of time trying to develop plant viruses as a model for understanding genetics in macroorganisms. That was, I think, difficult. There's not a whole lot of sort of results that came out of that. But I think it was sort of like a fruitful experience. The next stage in his career was when he began as a fellow at the Institute of Molecular Biology at the University of Oregon in 1960. And there he continued to work on T4 phages, trying to understand, again, what the relationship was between DNA and RNA. So one of the things that he did was he demonstrated a circular linkage map. So the chromosomal structure of these T4 phages is pretty complicated, and they'll undergo circular permutation. But he did some work trying to understand that, and that was important for understanding that the sort of circular structure of DNA was commonplace within microbial organisms. And he also did work on understanding what the code was relating DNA to a protein, again, by studying single nucleotide mutations within the T4 phage. But in the back of all of this, he was thinking about the need for a new model system. And so at this time, the phages has really sort of become this sort of important model for understanding genetics. But people recognized the limitations of working with phages only and saw a need for developing similar types of techniques and tools in macroorganisms. And folks were working on Drosophila and kind of developing these tools in Drosophila, as well as C. elegans, both phenomenal model systems with a lot of their own advantages, which hopefully have been talked about in this series. I've also worked on Drosophila, so that particular model is near and dear to my heart. But Streisinger saw a need for a vertebrate model organism. And his reasoning was that invertebrates and vertebrates had diverged quite a long time ago. So this is a phylogeny, so a family tree for animals, showing the sort of common ancestor of invertebrates and vertebrates here, 990 million years ago. And the C. elegans and Drosophila sort of branch off. And then there's, it's not until 450 million years ago that you see the divergence of fishes from tetrapods and other vertebrates. And so what he was looking for was a vertebrate model organism where it was possible to apply these genetic techniques, with the hope being that because of the sort of closer relationship to humans, that there'd be a more direct relationship, a more direct generalization of the conclusions drawn in that model to humans, so that it would be possible to kind of leverage those tools and techniques for biomedical applications. So what he did was he went looking around, kind of thought about this. Hopefully I read the paper by Creaser, and ultimately came across zebrafish as a model. He got his first batch of zebrafish from a pet store in Albany, Oregon. They're still a very common Aquarius, still a very common species to find in pet stores. If you go down to the Petco that's next to my house, you can still get zebrafish. And he thought, oh, that looked like a great system for a lot of these same reasons. And so then he started working on developing that into a real genetics model organism. There's lots of funny stories there. He was able to get a small hut built next to the building that they were working in. And they would set up portable heaters to try and get the temperatures at the right level for these tropical fish species in Oregon. This part of Oregon, I've lived in this part of Oregon, it doesn't get that cold, but it's not good weather for tropical fishes. It's not 28°C year round. And so sometimes the heaters would break, and then all the animals would be lost, and they'd have to start all over again. And then you had to figure out how do you, not only how do you rear them, how do you hold the adults, how do you get them to spawn, how do you collect the embryos, all of these sort of logistical challenges of working with these animals. And so it's a sort of slow simmering project that happened in his lab. And you can actually see that. I managed to get a copy of his CV for this period. And so he started at Oregon in 1960, and I think very shortly after started sort of like working with zebrafish as a model system. And the first paper using it was in 1981. So 21 years elapsed between when he started the position and started sort of tinkering with the system and when the first paper that came out of that work, to the best that I could sort of dig up. But that paper was a doozy. And so this is the first paper that he put together on zebrafish. And it's production of clones of homozygous diploid zebrafish. And the naming here is the genus has changed a little bit as the information has come in. So we now refer to it can be referred to either as Brachydania or Danio because the genuses have been merged. But what he found was that it was possible to produce a diploid clone of a zebrafish. And the technique that he used was to take a unfertilized egg and then take a sperm that had been essentially neutralized by exposure to a UV so that they could sort of initiate the developmental process but would not contribute DNA. And so then he would allow the sperm to fertilize the egg. And then what he would do is apply a combination of pressure and ether that would inhibit that first cleavage. So the DNA would undergo replication, and you would get 2 copies from that the starting haploid material. So you normally have 2 chromosomes in pairs. The egg has 1 copy, the sperm has the other copy, and that's how you get the diploid organism. So he took the oocyte that has 1 copy, he took the sperm that had 0 copies because it had been neutralized, and then he allowed the oocyte to undergo division, or to replicate the DNA. But he prevented the cell from actually breaking into 2 parts. And so instead of that 1 copy being split into 2 cells, the 2 copies remained in a single cell. And so he got a single cell with a diploid copy from the one mother. And then from that, he's able to sort of grow that individual up, and he gets a clone of the organism. And this was widely reported within the popular media. People got very excited. So this is a cartoon from the Chicago Tribune, the same year that that paper was published. And so I guess this is that's supposed to be Streisinger coming home saying, you cloned him, and then his spouse is saying, you cloned him, you clean him. And so it really takes off at this point, not just sort of within the popular imagination, but that sort of press attention also brings a lot of attention to zebrafish as a model system. A lot of people got excited that these tools and techniques that you could use for trying to understand how things worked in invertebrates could also be applied really for the first time to a vertebrate model system. And so, for example, one of the this is sort of a beast of a figure, but I'll go through it a little bit. And so one of the things that you can do because of the features of the system is 4 genetic screens. Now these have been fundamental for work in Drosophila and C. elegans, and for the first time can be applied in a vertebrate model. And so the process is very similar as you would see for a fruit fly or for a nematode, where you have, or the principles are similar. You have a mutanized male that then is crossed against a wild type. This produces offspring that have some of those mutations, and then these get crossed to each other, eventually resulting in offspring that have 2 copies of the mutation. You can then see that mutation, and you can understand that this particular gene has produced this particular phenotype within the animal. And the advantage of doing this in a zebrafish is the scale at which you can do this. You can, in principle, do this in any organism. But if you look at some of the numbers that are required here, in this case, there's 266 males. You wind up with 3,000 families. Each of these is in a separate tank. Then you do 11,000 crosses down here. There's 500,000 embryos. So this is the point where you're really grateful that every single time you put embryo or 2 parents together, you get 200 embryos. And you're really grateful that you can keep animals in tiny dishes so that you can keep tens of thousands of different genetically independent lines in a room that's about this size, as opposed to a room that's the size of a very large conference center. And so it's really and you're really grateful for the fact that the animals reach maturity, reproductive maturity at around 3 to 4 months, so that you can go from one generation to the next over the course of months rather than years or decades. So at this point, thousands and thousands of different mutations have been generated, and each one of those provides a little window into the genetic machinery by which development occurs. And so I want to run through like one little example on kind of how that process works, how you use a mutant in order to understand some of these different developmental pathways. And so a normal developing zebrafish looks like this. So you have the head and then what's going to the tail over there. And one of the things that's important is the development of dorsal ventral, what's called dorsal ventral axis patterning. And the idea there is that if you're a little cell somewhere in, let's say here, you're a cell right there, I can look at that and I could say, oh, well, that cell's just behind it's at the back of the head. I know that because I'm way out here and I can look and I can see where that cell is. But that cell has no way of knowing where it is in the universe. It has no way of knowing whether or not it's near the head or whether it's near the tail. It's a cell. All it can see is, it can't see anything. It has no eyes. It's a cell sort of sitting in the tissue. And so the way in which that cell can sort of identify its location within the organism is through the diffusion of small molecules and the surrounding tissue. And so this is a very old idea within developmental biology, where you have what are called morphogen, these small chemicals being sort of released at different parts of the embryo. They sort of diffuse outwards, and then they kind of those chemicals tell this cell where it is within the body. And you can go to like finer and finer different gradients in order to understand your position within the organism as a whole, within tissues, etc. But at this stage, it wasn't really well understood. So we have the sort of classical developmental biology telling us there are chemicals that diffuse, right? We don't know what those chemicals are. We don't know what the receptors are. It's, it's a very sort of like a vague understanding of what's going on. But these mutants allow us to get at that. So for example, one of the mutants that was identified is a mutant in a gene that's called BMP2, which doesn't really matter what it stands for, but it's bone morphometric protein. And when you knock out this specific gene, then what you find is that embryos that look like this. It's a little bit hard to tell, like if you're not used to looking at zebrafish embryos, what that is, but it's an embryo that's all head, basically. Is that embryo is just one head and there's no tail. It's a little bit more clear if you add in a little bit of BMP2 back in. So we've fully removed BMP2 and then we're just going to add a little bit back in. And what you find is that you get recovery of the sort of tail features. And if you add even more BMP2 back in, then you get even more tail. And if you add a whole lot of BMP2, then it's all tail. And so what that's telling you is that this BMP2 is the presence of that tends to signal for tail. So that if you have more BMP, then you're telling the cells that that it's more oriented towards the tail than towards the head. And if you do lots and lots of those experiments, then you can build up an understanding for exactly what these chemicals are that are generating these different gradients. And so the sort of current understanding is that there's a large number of different BMP producing cells at the tail side of the animal. Within the development, we would call this the ventral side because development is complicated and things kind of fold around a little bit. But for our purposes, that the tail side is going to be what we would call the ventral side of the embryo, and the head side is going to be the dorsal side. So we get a large number of BMP producing cells at the tail side, and some cordon, which is another chemical, producing cells at the dorsal side, and they're wired together in this sort of complex circuit where cordon is inhibiting BMP, and then BMP inhibits cordon, with the effect that you have this gradient between BMP and cordon. So if you're a cell and you have a receptor out in the universe and you're seeing lots and lots of cordon, what that means is that you're near the head of the animal, and you should differentiate into head cells. If you're a cell and you are looking out into the world and you're seeing lots of BMP, then that means that you're near the tail and that you should differentiate into tail-like cells. And so this was really important because it is a gateway for understanding development, but also for biomedical applications, understanding different disease states. You can do further studies to sort of understand the downstream molecular pathways, and in this case, the BMP pathway is mediated by BMP that's in the environment, and then binds to receptors on the surface of the membrane, and the effect is that then activates downstream proteins that'll then bind to the DNA and activate certain genes. And so the idea is that you have these BMP molecule floating out in the environment. It's recognized by the cell, and then that turns on other genes. And from there, you can do, from there, the cells can do all kinds of complicated things. They can turn on genes that would produce opsins, then they can develop into eyes or retinal cells. They can turn on genes that would turn on other receptors that would make them sensitive to other compounds. They could turn on genes that would produce other morphogens that would do other types of signaling. You get this sort of large regulatory networks that produce the full sort of spread of different cell types and the full spread of complexity. That was really the start of the model system, was understanding, using these genetic tools to understand different developmental processes. But since then, zebrafish has developed to grow as a model system, and it has been leveraged as a model system for a number of other fields, including toxicology and neuroscience and drug discovery. And part of that is the is the benefits that we talked about before, is that they're small, they work well in a laboratory environment. You can get lots of embryos at any given time. And part of it is also the status as a model system makes it great as a model system. And so when people have done this sort of work to understand the anatomy, that becomes a tool that you can then leverage for other studies. And so there's a sort of synergy, sort of multiplicative effect, where once people have worked out what are some of the basic gene regulatory networks, then when I do a study and I see an effect, then I'm better able to interpret my own data. So even in toxicology studies that may not be asking exactly the same questions, they're still able to leverage all of that kind of knowledge base in order to understand their own results better. They're able to use the understanding of the genes for studying the anatomy, for studying how their compound might affect some of these other pathways. You'll notice at this point I have mentioned nothing about the natural history of the organism. And that's in part because this talk is done sort of chronologically. This is really the part about the sort of period in the field where people started to look around and go like, where did these fish even come from? Sort of early 2000s, after zebrafish really started to become successful as a model, people started to think about like, where did these, they came from a pet store in Albany. Where were they before that? And so people started to think more seriously about this. There were some expeditions launched to do surveys, trying to understand the sort of natural habitat and the natural history of these organisms. And so the zebrafish come from largely freshwater rivers in the northern part of India, on the border between Nepal and India and Bangladesh and India. And so every one of the little dots shown here is a sort of observation of zebrafish in the wild. And they tend to be found mostly associated with sort of rice production in some of the standing water near the rice fields. And so here's them seining for zebrafish and finding zebrafish in this body of water. They're also found in some slow-moving creeks. And they tend to be found in both the sort of sunnier patches as well as in the little shaded areas. It's perhaps this habitat that has made them a good model system for us within the laboratory, because these small bodies of water tend to have highly variable water quality conditions. The temperature can change very rapidly because it's a small body of water. Different soil conditions can cause different pH values, et cetera, et cetera. So the animals have evolved to live under very variable conditions. They're also omnivorous. And again, this is a feature which is kind of unknowingly helped us within the laboratory environment. And so they don't specialize on a particular prey item. They'll eat anything, essentially. They eat a lot of aquatic larvae of terrestrial insects. These are mosquito larvae, which they're thought to eat. They'll eat algae. They'll eat microcrustaceans. They'll eat other types of detritus that would be found within the water. They'll eat more or less anything, which again makes it easy to develop, to rear them in a lab environment. They tend to, in the wild at least, they mate just before the monsoon season, typically at dawn, and they perform a nice little courtship dance. So here's a male and a female courting. Oh, guess who the male is and who the female is? So the male is this one. So the courtship dance is going to be chasing the female. Watch that again. So it's going to consist of chasing the female and then trying to, generally in a larger environment, kind of lead her to the spawning grounds. And then the male will also, will sometimes perform like circling. So if the spawning ground is here, then he'll kind of circle around between the female and the spawning grounds, sometimes circle around the female, but generally sort of like circling and chasing and try to lead the female to the spawning grounds. And then if he's successful, then the female will kind of follow him and eventually spawn. When she spawns, she'll produce, again, as similar to a lab, hundreds of embryos, typically not over the course of like multiple encounters. So maybe 20 embryos per encounter, and then over the course of a morning, maybe 200. The eggs are non-adhesive and then sink down to the bottom. They'll hatch after around 3 days, and then the larvae will attach to the substratum. They have sort of adherent cells at the bottom of their head, and then after around 5 days, they'll inflate their swim bladder and sort of swim off and then be on their way. They have a number of natural predators within the habitat, including eelfish and snakeheads, and then featherbacks, and then also other zebrafish. And so I don't know if you noticed in that dish, we have sort of a perforated bottom on the dish. That's because we need to protect the eggs from the parents. What'll happen is the parents will lay the eggs, swim around. By the time they come back, they've forgotten what those are, and they'll eat them. And so, yeah, it is thought that the zebrafish will prey on the eggs. In the natural habitat, that's probably fine because there's so many eggs from so many other individuals that if you find an egg floating around, it's probably not yours. But in a lab environment, yeah, we need to protect the embryos from the parents. They also tend to live in groups and exhibit shoaling behavior. So here's an example of that. So in an environment where there's lots of predators, a few individuals wander that way, it's gonna realize he's all alone, doing his own thing. Probably should join the group. Yeah, and so again, as a mechanism for predator evasion and everything else. And so it's an organism which, again, kind of started as a organism that was used for as a genetics model system, where the focus was on development. But of course, it's an animal that evolved in a natural habitat and which has a rich repertoire of different behaviors. And so some of the current work in zebrafish is looking at those behaviors and trying to understand some of the neural circuits that are responsible for those behaviors. And so that's what my lab works on, is that we use zebrafish as a model system for understanding the neural circuit structure and organization. And we don't actually work on the adults. We tend to work on, it's a little pixelated, sorry, but we tend to work on the larvae. And the larvae have a number of advantages for us. They have, if you look at a larval brain, it has a sort of stereotypical vertebrate structure with a forebrain and a midbrain and a hindbrain, especially within the midbrain and the hindbrain. We see a lot of homology to the brain of other vertebrate organisms. But within a larval zebrafish, you have all of those structures in the sort of smallest, most miniaturized form. So instead of having millions of some particular cell, we might have a couple of dozen. And so the entire brain fits within this sort of imaging volume of a microscope. You can see the entire brain with cellular resolution all at once. And so that really poses a really sort of fantastic system for trying to understand how some of these more complicated circuits within the central nervous system work. If you were to look at just approximate number of neurons, which is sort of a crude metric for neural complexity, but is maybe somewhat enlightening. Humans might have 90 billion. By the time you get down to a larval zebrafish, it's 100,000. And so it's a very small, sort of as close to a tractable vertebrate model as you're going to get. And here's what the brain looks like, again, with a forebrain, midbrain, and hindbrain. There's a cerebellum over here. Right. And so what my lab in particular looks at is trying to understand neuromodulatory pathways and trying to understand how different stress states affect the behavior of the animal. And so, obviously there's not necessarily a direct relationship between the sort of emotional state of a larval zebrafish and the emotional state of a human. But what I hope to convince you of is that a lot of human behaviors build on top of very old, very fundamental animal behaviors. So for example, it's Florida, it rains a lot here. Imagine you're running, to your car, and all of a sudden you just, you get this sort of downpour that comes out of nowhere. What does everybody do? Like throw something on your head and you make a run for it, right? You try and get to your car as soon as you possibly can. You try to avoid the rain. And what if you realize like your car is a mile away, you are not getting there anytime soon? What is your response? Exactly. It's yeah. You're just gonna live with it. The rain is not gonna kill you, and so that actually reflects an ancient shift in behavior, where we would call that first response active coping. And so, in response to a acute aversive stimulus, most animals will display what's called active coping or avoidance behavior, is get away from the thing that is aversive. But prolonged exposure to the same exact stimulus, provided it's inescapable, typically results in what we would call a passive coping or a sedentary behavior, where you just hunker down and wait for that stimulus you just ride it out and wait for it to go away. And so one of the first questions we asked was whether or not our larval zebrafish, 5 days old, 100,000 neurons, will display that same type of behavior. So we did a very simple experiment. We put a few animals into a dish. We filmed them. We have some computer software that we wrote that'll sort of track the animal as they're noodling around inside of the dish. And then we expose them to a mildly noxious stimulant. This is mustard oil. So it's not going to hurt the animals, but they're not fond of it. They'd rather avoid the mustard. And what we find is that they display exactly that same type of active coping response that you would expect from other organisms, where there's this immediate increase in swimming speed as they try to escape that stimulus. But when they're unable to do so, within a couple of minutes, they shift to a sedentary behavior. So it's actually their swimming speed decreases below their sort of baseline activity. So it's not that they just get used to it. They actually they're more sedentary than they were when they started. And so the advantage of larval zebrafish is that we can then use some of the genetic tools to go back and try and understand at a cellular level what's going on. Where are the neurons that are mediating this response? And so we use engineered fluorescent proteins that will glow when a particular cell is active, and then are dark when the cell is inactive, so that we can use optical techniques, we can use microscopes to record the brain of the animals and look at which cells are responding and which ones are not. And so the paradigm that we use is we use engineered proteins, or the DNA for engineered proteins. Those are injected into cells. We take our little larvae and we immobilize them so that they can't swim away. They can still move their tails, so they think they're swimming around very happily, but we can sort of hold them still so that we can image from the brain. We use a multiphoton microscope that allows us to image deeper into the brain that would be possible using a traditional microscope. And we use machine learning techniques to identify specific cell types that are mediating the response. So I'm going to give you an example of what that data looks like. So here's baseline. This is a projection of the brain. Because it's a projection of a 3D structure, I've sort of color-coded the different neuron populations. So if you see a change in brightness or in color, that reflects a change in the activity of the neurons. So that was baseline activity, and then mustard is being acted. Let me go back and show you that again. Oops. Okay, so this is spontaneous activity of the animals. Animal's not doing anything. There's no stimulus. It's just sort of sitting there, kind of thinking its innermost thoughts. And then when we add mustard oil to the environment, we see a shift in the activity of this population, and then a shift in the activity of these populations up here. These are associated with olfaction. And then we also see like a number of other neuron populations change activity. We use machine learning to figure out which of the neurons is kind of most strongly correlated with the change in the stimulus. And what we identify is a group of neurons in the back of the brain here. And each one of these little dots, it's a little pixelated because the animation, but each one of those little dots is a individual cell. And so we can record the responses of the animals at cellular resolution. We see every single cell within the brain of the animal as it responds to the stimulus. And we can then look at not just this part of the brain responded to the cell, but we can say this individual cell responded to this stimulus, but not to this stimulus. This individual cell responded a little bit ahead of this other individual cell. And so we can look at 100,000 cells in parallel and start to piece together exactly how the circuit is put together. And so this is just a little bit of additional analysis showing that even within that area postrema, there's two different populations, one which has a sort of faster response and one which has a slower response. And the transition between them actually matches what we see in the behavioral transition. So we think that that kind of shift in relative activity between the two populations may be causally related to the shift in the behavior. And so, as you can see, hopefully, it's a powerful tool for understanding how the different neural circuits are put together and how trying to understand how the neural circuits within the vertebrate nervous system interact with each other in order to produce more complex behaviors. I'll skip that. And then I'll real quickly go through a couple of one last project that we recently started. So we've been looking at kind of understanding how the neural circuits work, but recently we've been trying to understand, can we use this as a tool for understanding new compounds whose activity we don't understand yet. And so we've partnered with the Luskin lab, that's a drug discovery lab, and we've started using zebrafish-based behavioral assays to identify new compounds that ablate the response to noxious chemicals, and then whole brain imaging in order to understand the mechanisms by which they're doing that. And so here's some specific data where these are different samples. These are different sort of potential drug leads. And each one of them is tested in a behavioral assay. So black in this figure is a no movement. Copper would be a high movement. And so you can see that sample 2 and sample 3 have substantially reduced the effect of this animal to the sort of stressful stimuli. And then subsequent work can identify the mechanism of action, again, using techniques like whole brain imaging, and then individual studies on individual cells expressing individual receptors. All right, and hopefully with that, I've convinced you of the value of zebrafish as a model system. And I'd like to thank my many collaborators. People in my lab, like Austin and Summer and Bushra and Brandon and Monica and Amelia, who have been, who have basically done all of the data collection. I'm just helping for the most part, as well as my collaborators in the Luskin lab and sort of ongoing collaborators at the Martindale lab. And I'm very happy to take any questions.
Okay, good. Great, thank you. Questions? Okay, Ken.
This is more of a comment.
Okay.
In 1960, I went to the phage meeting, Cold Spring Harbor. George was there, Max Delbruck was there, Luria, Hershey, all the guys who shared the Nobel Prize for the phage and the origins of molecular biology. But Delbruck was kind of a standout guy. And the meeting is very informal. Everybody's sort of dressed as we are, except for George, who is actually wearing a bathing suit and a sweatshirt and some sandals. And he is kind of an edgy guy. He was sort of marching around the stage, starting to give his talk. And Max stood up. He said, "George, I'd like to interrupt you at this point. to say, one, you're doing much better than you used to do as a graduate student. And two, I have a question for you. And George said, before you ask your question, Max, let me tell the audience what you were like when I was a student. And he told about how Max would call in a student after a seminar, and you didn't know quite what was going to happen. He'd either come down on you hard, or he'd pull out a bottle of scotch and you could have a drink together if things had gone well. But it was a very stimulating environment, I could tell you. And those guys were a lot of fun to be around. Even though I was sitting there, I was basically an undergraduate. So I enjoyed it thoroughly.
Yeah. Yeah. I think, I mean, part of putting together the slide, I tried to do a little bit of reading. I heard that the Cold Spring Harbor meetings were really, like, you look at the attendance list and it's like Nobel Prize, Nobel Prize. Oh, sure.
Can I ask a question while she's doing that?
Absolutely.
When they were seining in India,
Yeah.
is it for a study or do they eat them? What were they collecting them for?
Oh, so that was for the study. So they were trying to understand the diversity within the river, and part of it was also there'd been some informal studies before that, but part of it was trying to understand Danios in particular, because the taxonomy is confusing and there are different species that were being confused with each other. And so there were a couple of specific trips back there to try and understand, like, where are zebrafish in particular.
Okay, there's a question.
The picture you showed of all the tanks, that was a huge number of tanks of fish and eggs. I wonder why the big number, but also can you comment about the effort it takes just to maintain those? I don't know what they feed on and maintaining those. You had a number of pictures of people there.
Yeah.
Do you have the large numbers to look at rates of mutation, or why the large numbers?
Mm-hmm. So the
Can we go back?
sure. So the large numbers depends on the kind of the facility and what you're trying to accomplish. But one of the main reasons, main places where large numbers can be helpful is for like for genetic studies, where can you go back one more? So each one of those is a tank, actually. You can see in this one, each one of those is a tank with maybe 10 individuals. And that would be a fairly good sized zebrafish facility. Our facility, we have 3 of the racks, which is still a fair number of animals. I think I can go back. Okay, so the large number of animals comes from this experiment. And so the challenge with this type of experiment is that you don't know what you want to keep until you get to the end. And so you start off with 266 mutinized males, and then those get crossed here, and then those get crossed again, and you don't realize it's hard to figure out which of these individuals you want to keep until you're down here, basically, because you have to keep them all. And then you get here and you figure out, oh, well, this one was good, so we'll keep that one. And then those get isolated. As you can see, I think somewhere in here, so 500,000 embryos, so 10,811 F2 by F2 crosses. The screening happens with this generation. And it's not until here that you figure out, oh, well, 577 of those were good. So you had to keep 10,000 different strains. And each one of those 10,000 strains has to be a different tank, because otherwise you have no way of finding those individuals ever again. If you just sort of like throw them back into your tank, they're gone for good. So you have to have 10,000 different tanks, not necessarily all at the same time. So you can spread it out over, maybe a few different cycles. But lots of different tanks so that you can maintain the individual lines. In our colony, we're not doing for genetic screens, but we use, I mentioned having the genetically modified animals. And so we have 20 to 30 different genetically modified lines that we use for doing our stuff. And each of those has to be maintained independently. And we need a little bit larger number because they're stable lines and everything else like that. But yeah, it's maintaining the different genetic lines.
Was there work done at Whitney Lab on zebrafish before you entered into the picture? Did Dr. Lau have anything to do with that?
Yeah, definitely. And so the Liao Lab has been working on zebrafish for a number of years. And they're also interested in kind of neuroscience and those types of things. I think in the last few years, what we've been working together on a few projects and trying to kind of, I mean, the field as a whole is going on. So they were already moving in this direction and using these approaches. But what we've been trying to do is like more whole brain imaging and those types of things, using more genetic lines to kind of ask different questions.
And how big is an adult zebrafish?
Oh, that's a question I should have answered very early on, but didn't. They're about this big.
Yeah, I've seen them in the aquaria. They're just.
Yeah, they're little guys.
Any more questions? Okay.
Which is very convenient because if you need, again, tens of thousands of them, you don't want tuna.
Several comments. I found it fascinating. I never really could see any value of studying fruit flies, but now that you showed me the chart, I understand a lot more. Are zebrafish found just naturally in freshwater lakes and ponds?
So they're in India, and so they're widely distributed throughout the rivers in the northern India, but they're not found in the United States, and they don't really appear to be invasive here either. And so there was an initial sort of batch that was brought to Europe in the early part of the 1900s, and then that was brought to, I think it's actually in Florida. Again, we don't really know where the pet store in Albany in the 1960s got their fish, but it was probably actually a fish importer in Florida sold them to the pet store in Albany, and then Albany is where the founder population actually was for most labs.
So that explains why in Albany, because I was surprised, that's actually closer to Oregon State than University of Oregon.
Yeah, I think I don't know why he went to Albany, but yeah, so Oregon State is Corvallis. I was a faculty at Oregon State, and then University of Oregon is in Eugene. I don't know why they went to Albany. Better pet store, maybe.
It's curious that they're not invasive. I mean, guppies are, and all kinds of other, particularly here in Florida, all kinds of other pet store, aquarium fish are certainly like tilapia, for example, and all the cichlids. They're very invasive, but this is not, huh?
No, it doesn't seem to be. It's not clear why that is. I mean, part of it might be, to speculate wildly, is that these have been reared under artificial conditions for, since the early 1900s. So and they would breed maybe once a year to kind of propagate. So that's a few generations. And there's no predators when you're rearing in our lab. And so there's selection for reproductive capacity, but not necessarily for like predation evasion and those sorts of things. So there are some questions like how much do the behaviors that we see in the lab reflect the behaviors that would be present in a wild population? And it's possible that our lab reared individuals are just they're basically just predator food in the environment. That's speculation, but yeah, it could be those types of factors.
And are they, the way you rear them, is there mortality in your rearing system, or are they, in other words, do you have to be really careful with them when you're rearing them and give them lots of clean water? I mean, there's other things like that maybe disease would get them.
I think generally the mortality is pretty good. So we would it depends On so they, like many fishes, they have a window of time where you have very high mortality. It's just as they begin. So once the yolk sac is gone, so they can live off the yolk sac up until around 10 days. And then once that yolk sac is gone, they have to start feeding themselves. And if they don't start feeding themselves, then by 12 days, they're not going to be there anymore. And so there's this lethal window, and that's consistent for zebrafish. It's actually consistent for most fishes, is that there's a window of high mortality once the yolk sac runs out. And so we do see that where we have to make sure that we're giving them lots of available food at that window. But if we do that, then we get survivorship typically in the 80 percentile, 80% range. So it's probably much better. Well, it's certainly, they make 200 eggs. So it's certainly much better than they would in the wild. But one of the things that we do have to worry about is that we're working on the larvae. And so we're working on stress. And so we take very, very good care of our animals because we want to make sure that our baseline is not stressed. So the water quality and everything else is very, 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 very carefully monitored to make sure that they're in absolutely pristine conditions so that when we do apply a stimulus, it's not on top of some weird background. We don't use seawater. So we use, actually, for our water, we use highly purified DI water, and then we add back salt and buffer and everything else. So we start with sterile water.
Yeah. Yeah. Okay, any other questions that anybody have? Anybody on Zoom? No? Okay.
Worked at the Whitney lab, and he was at the other extreme. The fish he worked with were sharks, and to see Bill carrying around a shark, thrashing, but at the same time, there was a member of the biology department who discovered or concocted something called gator bait. Gator bait contained all the essential amino acids that fish found tasty in their food. And they could get the fish in their tanks, including the sharks, to do quite amazing things when they added some gator bait to the system.
Yeah. Yeah, I mean, there's another faculty. So I work on zebrafish, but also started working on some other organisms as well. So we've got a project looking at ctenophores, this little gelatinous zooplankton. That's been fun because, so they're a group that diverged from the rest of all other animals very early on in the evolution of animals. And so working on ctenophores, which is, very much, we're trying to make it a model organism, but at present it's not. And zebrafish has been kind of interesting because you can draw a lot of insights. There's also another faculty at Whitney, Leonardo Ibarra Castro, who's doing work on aquaculture, really trying to raise fish for like food production. And and he's done some really amazing stuff. And it's funny to look at his operation versus mine because we rear clutches of 200 embryos and we think we're doing fine. But he'll raise clutches of 20,000 in big like swimming pools. It's different. We don't really need 20,000 embryos for our research. So it's different approaches for different questions. But yeah, it's funny to see like the different scale of operations.
Okay, well, thank you so much. Really appreciate your insights and it's just great telling us all about these model organisms. So thank you.
Thank you for inviting me.